Why do identical twins have different genetic disease risks? Have you ever wondered why identical twins, who share nearly all their DNA, can have different health outcomes? It's a fascinating question that dives into the world of genetics and health. Let's look at the reasons behind this phenomenon. First, while identical twins come from the same fertilized egg, they are not perfectly genetically identical. After the embryo splits, small genetic changes called mutations can happen in one twin but not in the other. These mutations can affect genes that relate to disease risk. So, one twin might develop a condition while the other does not, even though they share most of their genetic material. Next, we have epigenetics. This term refers to chemical changes on DNA or proteins that regulate how genes are active without altering the DNA sequence itself. Environmental factors and life experiences can influence these modifications, leading to differences between twins. For instance, twins may share some epigenetic patterns, but they can also have unique differences that affect how their genes are expressed. This can result in varying susceptibility to diseases like type 1 diabetes, schizophrenia, cancer, and autoimmune disorders. Environmental influences and lifestyle choices also play a big part. Even though twins may have similar early life exposures, their individual environments can vary. Factors like diet, infections, stress levels, and personal behaviors can differ significantly. For example, if one twin has a genetic predisposition to high blood triglycerides and also follows a high-fat diet, they may develop health issues while the other twin does not. Additionally, the shared uterine environment during pregnancy can impact disease risk. Research shows that factors unique to the twin pregnancy, like competition for nutrients, can affect birth weight and early development. These early experiences can influence health throughout life. From the perspective of family genetic testing and hereditary health, it's important to understand that identical twins can have different disease risks. This highlights the need to consider not just inherited DNA variants but also epigenetic factors and environmental exposures. When interpreting genetic testing results, it is essential to remember that identical twins may not have the same health outcomes. In essence, identical twins have different genetic disease risks due to postzygotic mutations, epigenetic differences, individual environmental exposures, and developmental factors. These elements combine to create unique health profiles despite their nearly identical DNA. Understanding this complexity is key when discussing hereditary and complex diseases.